Hello, my name is Kirk Balderas. I am a junior biology major, and I came to Texas Tech University through the NIH's Plains Bridges to the Baccalaureate program. And I'm currently working in the Nutrigenomics, Inflammation, and Obesity Research Lab under the mentorship of Dr. Naima Maustad Musa, Dr. Yujiao Zhu, and Dr. Kimbra Albrecht Schulte. And I'm presenting my research project on the hepatic effects of fish oil in male mice with UCP1 deficiency at thermoneutrality. Obesity is growing more prevalent every year in the United States, and data by the CDC shows that every state in the US has an adult obesity rate above 20%, with the country's average in 2018 being 42%. So what is obesity? Obesity is described as having a body mass index of more than 30, and it's important that we understand how to deal with obesity because it has many health-related risks, including heart disease and stroke, type 2 diabetes, and some cancers, including breast cancer, liver cancer, and colorectal cancer. But the main risk we're examining is non-alcoholic fatty liver disease. Fatty liver occurs when fat begins to accumulate in the liver at a rate of more than 5%, and this leads to liver steatosis. This is a major risk factor associated with obesity because more than 70% of people suffering from obesity will develop fatty liver. And liver steatosis is caused by the accumulation of lipids within the hepatocyte. Liver steatosis is relatively benign and reversible. If untreated, this can progress to the next stage called steatohepatitis or NASH for short, and this leads to cirrhosis and inflammation. And this is why liver disease can lead to such high levels of mortality. Liver steatosis is treatable before the levels of liver damage become too great, and some treatments for steatosis include lifestyle, including diet and exercise, drugs that reduce fatty liver, or dietary supplementation with things like fish oil. The reason fish oil is so effective is because it's rich in omega-3 fatty acids, one of which being eicosapentaenoic acid, or EPA. And EPA is the fish oil of choice in this study because of its anti-inflammatory properties and metabolic benefits. Other common health benefits of fish oil include the reduction of triglycerides, and it can reduce inflammation. Fish oil is able to do this through reducing inflammation and improving lipid metabolism, which reduces fat accumulation in the liver. To understand how fat plays a role in obesity, it's helpful to understand what kind of fat is present in the body and how they contribute to obesity and therefore contribute to non-alcoholic fatty liver disease. And the most common type of fat is white adipose tissue. And this typically is referred to as the bad fat. White adipose tissue stores extra energy. It's larger in size than other types of fat, and it contains few mitochondria. Many of the negative side effects we see come from the inflammation of white adipose tissue. The second type of fat present is brown adipose tissue, and brown adipose tissue is considered good fat because it is smaller in size and is more effective at releasing excess energy. Unfortunately, humans do not possess a significant amount of brown adipose tissue, and overweight humans have this as a limitation. Brown adipose tissue appears brown because of the large number of mitochondria it possesses. And what's special about these mitochondria is that they possess a unique protein called UCP1 or uncoupling protein 1. Uncoupling protein 1 uncouples the electron transport chain, which is an important metabolic process, and this creates energy in the form of ATP. And UCP allows for the free flow of protons across the mitochondrial membrane, and this generates heat in the form of non-shivering thermogenesis. The study design for this project begins with taking mice that are lacking in the UCP1 protein and feeding them a high-fat diet with or without the EPA fish oil supplementation for 14 weeks in a thermoneutral environment. And the thermoneutrality was used to prevent the interference of shivering thermogenesis. After 14 weeks, tissue samples were collected for analysis. Once liver samples are taken, RNA isolation is conducted so that RNA sequencing can be used to analyze the transcriptome of the mice. Using a program called Ingenuity Pathway Analysis, or IPA for short, we are able to compare how genes are differentially expressed in test mice to a known genetic standard. IPA also allows us to see how these differentially expressed genes are involved in different metabolic pathways. Our objective in this study is to determine how gene expression is affected by the use of EPA in the absence of UCP1. And our hypothesis is that when EPA is combined with a high-fat diet, it will increase the presence of genes involved in pathways such as beta oxidation, thus preventing or reversing the effects of non-alcoholic fatty liver disease. When we take a look at how EPA affected body weight, we can see that there was only a minor difference between the body weight of the two groups. So at the end of the study, they were relatively close. But the truly significant difference was with the concentration of hepatic triglycerides between the two groups. EPA was able to significantly reduce the concentration of triglycerides in the EPA group independent of body weight. And looking at these H&E stains, you can see the physical difference between the groups, with the high-fat group containing much higher levels of liver droplets within the hepatocyte. So IPA is able to generate a list of genes involved in the pathways that are being differentially expressed. And it gives us a list in order of how significantly they're altered from the normal. And by looking at this list, we can see the top pathway that's being upregulated by the use of EPA is fatty acid beta oxidation. And this finding is important because we know that fatty acid beta oxidation is used to break down fatty acids and convert them to energy. So IPA generates a list of the genes involved in this pathway that are being differentially expressed by EPA. And all the genes that are being differentially expressed by EPA in this pathway are all being upregulated, leading to a higher expression. That leads to the upregulation of the fatty acid beta oxidation pathway through the higher expression of these genes. And from the list of genes generated by IPA, we can see how the beta oxidation pathway and how those genes are involved. And so here we see ACSL1, an enzyme which plays a role in fatty acid degradation, and ACAA2, which catalyzes the final steps of mitochondrial beta oxidation, and EHHADH, which plays a similar role in peroxisomal beta oxidation. And all of these steps result in products such as NADH, NAD+, and hydrogen protons that will be used in the electron transport chain and later create ATP for energy. In summary, high-fat diets lead to obesity and the accumulation of triglycerides in the liver. And this accumulation creates inflammation that causes non-alcoholic fatty liver disease. And all of this is exacerbated by the absence of UCP1. And if we supplement a high-fat diet with fish oil, we can use this to upregulate pathways that break down fats and prevent the development of non-alcoholic fatty liver disease. The impact of this research comes from fish oil's effectiveness at improving hepatic steatosis in the absence of UCP1 by upregulating hepatic lipid oxidation. And this has real life implications because Americans specifically have a national obesity rate of 42%, with 70% of those people developing liver disease. And human adults do not possess high levels of UCP1, meaning omega-3 fatty acids found in supplements like fish oil can be used to create or prevent the development of non-alcoholic fatty liver in humans in the future. Lastly, I would like to acknowledge my mentors and everyone who works in the Neora lab, as well as my mentors from the Plains Bridges program, Dr. Lacey Alexander and Dr. Kanyas Carroll, and the NIH NCCIH for funding this project. Thank you.